Genetically modified organisms. Genetically modified organisms can be performed in any type of organism such as plants, food, human, animals or bacteria. Let's see how GMO made.
How are GMO plants made? First, scientists look for a desired trait in a plant, animal, or even bacteria. It could be a trait like resistance to drought, insects, or viruses. Then they copy the gene that contains that trait and insert it into the DNA of the plant they want to improve. Scientists then grow that plant to see if it adopts the desired trait. Feed your mind with more GMO facts on FDA's website.
GMO stands for Genetically Modified Organism. Let's break it down word by word. Genetically refers to genes. Genes are made up of DNA, which is a set of instructions for how cells grow and develop. Second is modified. This implies that some change or tweak has been made. Lastly, we have the word organism. When it comes to GMOs, many people only think of crops, yet an organism isn't just a plant. It refers to all living things, including bacteria and fungi. With that in mind, GMOs are living beings that have had their genetic code changed in some way. While conventional breeding, which has been going on for centuries, involves mixing of all of the genes from two different sources, producing a GMO is much more targeted. Rather than crossing two plants out in the field, insert a gene or two into individual cells in a lab. Yet, as mentioned earlier, GM technology can also be used on microorganisms. For example, bacteria have been genetically modified to produce medicines that can cure diseases or vaccines that prevent them. A commonly used medicine that comes from a genetically modified source is insulin, which is used to treat diabetes, but there are many others.
The process to create a GMO starts very small. A scientist causes a gene to be inserted into the DNA in the nucleus of a single cell. The DNA being used for the modification is so small that it cannot be seen even under the most powerful microscope. Despite how tiny a cell is, there is a massive amount of DNA all packaged into its one little nucleus. To give some idea of just how much DNA is packed into that small space, if you were to take all the DNA of one single corn cell out of the nucleus and line it up end to end, it would be about 6 feet long. Into this enormous amount of DNA, a very small piece is inserted. A vast majority of the organism's genetic code remains completely unchanged by the process.
This single cell has been modified. The scientists will treat it with naturally occurring plant hormones to stimulate growth and development. This one cell will start to divide, which is the natural growth process for any organism, and the resulting cells begin to take on specialized functions until they become a whole plant. Because this new plant was ultimately derived from a single cell with the inserted gene, all of the cells in the regenerated plant contain that new gene.
Genetic engineering allows scientists to select one specific gene to implant. This avoids introducing other genes with undesirable traits. Genetic engineering also helps speed up the process of creating new food with desired traits. The possible benefits of genetic engineering includes more nutritious food, tastier food, disease and drought-resistant plant that requires fewer environmental resources such as water and fertilizer. Less use of pesticides, increased supplies of food with reduced cost and longer shelf life, faster growing plants and animals, food with more desirable traits such as potatoes that produce less of cancer-causing substances when fried, and medical foods that could be used as vaccines or other medicine. In research studies, animals that has been safely genetically engineered include cattle, pigs, chickens, goats, sheep, dogs, cats, fish, rats, and mice. And in plant, the majority of genetically engineered plants, including corn, canola, soybean, and cotton, are typically used to make ingredients that are then used in other food products.
Some people have expressed concerns about GE foods such as creation of foods that can cause an allergic or toxic reaction, unexpected or harmful genetic charges, inadvertent transfer of genes from a GM plant or animal to another plant or animal not intended for genetic modification, foods that are less nutritious. These concerns have thus far been unfounded. None of the GE foods used today have caused any of these problems. The U.S. Food and Drug Administration or FDA assess all GE foods to make sure they are safe before allowing them to be sold. In addition to the FDA, the U.S. Environment Protection Agency or EPA and the U.S. Department of Agriculture or USDA regulate bioengineered plants and animals. This assess the safety of GE foods to humans, animals, plants, and the environment.
Why do we have GMOs? Actually, we've been modifying crops for thousands of years to prevent crop loss from pest and weather damage, to grow more food on less land, even to improve nutrition. Today, GMOs are developed for the same reasons. With genetic engineering, scientists can change and improve crops more easily and quickly. Feed your mind with more GMO facts on FDA's website.
Now, let's give the meaning of each terms. First, the GMO or the genetically modified organism. It genetically refers to genes. Genes are made up of DNA, which is a set of instructions of how cells grow and develop. It provides certain advantages to producers and consumers. Gene therapy. It is a technique that modifies a person's genes to treat or to cure disease. It can work by several mechanisms, replacing a disease-causing gene with a healthy copy of the gene, inactivating a disease-causing that is not functioning properly. Gene theory. It states that lifeline is largely determined by the genes we inherit. According to this theory, our longevity is primarily determined at the moment of conception and is largely reliant on our parents and their genes. DNA, or the deoxyribonucleic acid, is a long molecule that contains a unique or genetic code. It contains the instructions needed for an organism to develop, survive, and reproduce. Cloning. It is a technique scientists use to make exact genetic copies of living things. Genes, cells, tissues, and even whole animal can be cloned. Gene. It is the basic physical and functional unit of heredity. It is made up of DNA. Gene splicing. It is a post-transcriptional modification in which a single gene can code for multiple proteins. Golden rice. It is a variety of rice produced through genetic engineering to biosynthesize beta-carotene, a precursor of vitamin A in the edible part of rice. Genome. It is the entire set of genetic instructions found in a cell. In a human, the genome consists of 23 pairs chromosome found in a nucleus as well as a small chromosome found in the cell mitochondria. Each set of 23 chromosomes contains approximately 3.1 billion bases of DNA sequence. Hybrid. The offspring of the two plants or animals of different species or varieties. Tissue culture is the cultivation of plant cells, tissues, or organs on a specially formulated nutrient media. Stem culture are the roots which different cell lineages arise and they exist in multiple forms.
To sum it all up, a genetically modified organism is an organism whose DNA has been modified in the laboratory in order to favor the expression of desired psychological traits or the production of desired biological products. A genetically modified organism is any organism whose genetic material has been altered using genetic engineering techniques. In addition to that, here are some facts about GMO. Number 1. Over 12% of global farmland grows GMO crops as of 2016. Number 2. The first GMO food to reach the market was a tomato. Called Flarb Sarv, Calgon developed and sent the tomatoes to the FDA for approval in 1992. Calgon stopped producing it in 1997, so Flab Sar's modification gave it longer shelf life. Number 3. Genentech used GMO bacteria to produce insulin for use by diabetes patients. Doctors give insulin injection to manage the disease, with the hormone previously taken from human donors, but in 1978 Genentech developed GMO bacteria with human genes to produce insulin with. Other methods for genetic modification include electroporation and microinjection. Microinjection is the simplest method of genetic engineering by injecting new genes directly inside a target cell. One of the most advanced methods of genetic engineering is CRISPR gene editing. CRISPR, or CRISPR, is actually a means with which certain bacteria protect themselves from viral infection. That will be all about genetically modified organisms. We hope you learned something about it. Thank you.